First of all, I'd like to do an acknowledgement of country. I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects for the elders past and present, and the elders from other communities who may be here today. So it's my great pleasure today to introduce Sofonias Tessema, who will present his PhD completion seminar. Sofonias came to WEHI in 2012 on a Melbourne International Research Scholarship. He had previously completed his undergraduate degree at Jimmy University in Ethiopia, and he followed this with a master's degree in biology at Addis Ababa University, in which he received a perfect 4 out of 4 grade point average, which was fantastic and a really good sign. After his master's degree, he worked as a lecturer for 3 years at the same university and also conducted his own research into malaria, which resulted in a few publications. So when I saw Sofonias' CV, I was really excited, and I was happy that he decided to apply to do a PhD in my lab. And he was successful in getting a scholarship, and so when he came to WEHI, he was actually really happy to be given the opportunity. But to be honest, we're very lucky that he came to us. Sofonias is one of the nicest, smartest, most hardworking and professional people I have worked with. His dedication to his work is second to none, as you'll see today, all of the hard work he's done. His PhD project has been extremely challenging at times, but he's overcome many obstacles to get the job done. And in addition to that, he's always really positive and willing to help others, even when he's incredibly busy, like at the moment, and he's helping other people still. And this has certainly made my job as a supervisor incredibly rewarding. And so I'm actually really happy that he's to have the opportunity to work with him. So the work that Soph will present to you today is a summary of his PhD project, in which he's managed to identify key biosignatures of immunity to malarial disease. And so without further ado, I'll let him take the stage. So thank you, everybody.
Thanks, Eliza, for the kind introduction. Okay, this is my completion seminar, and I'll show you what I've been doing for the last 3.5 years. My PhD is really focused on understanding biosignatures of natural acquired immunity, with addressing the challenges of genetic diversity in human studies from Papua New Guinea. And this was done under the supervision of Eliza, Ivo, and Diana. Malaria is a global burden with an estimated 200 million cases every year and about 600,000 deaths, which is equivalent to the total population of Tasmania, if you want to put it into context. Of these, 80% of the people who die due to malaria are children under the age of 5 years old. Here is a map, a global map of the distribution of malaria mortality, and most of the mortality is concentrated in sub-Saharan Africa. But outside of Africa, we have the highest burden in Papua New Guinea, which is the focus of my research today. There are 6 species of Plasmodium, which infects human. Of these 6 species, I'll be focused on Plasmodium falciparum, which causes about 90% of the mortality due to malaria. Here's a life cycle of the malaria parasite. Malaria has a very complex life cycle, but I'm not going through all these life cycle stages. But what I want to focus is when human beings are bitten by an infected mosquito, the parasite injects the sporozoites, which develop in the liver into merozoites, and these merozoites infect the red blood cells. So my PhD is focused on a protein which is expressed on the surface of the infected red blood cells. So here is a closer look at the blood stage life cycle of the malaria parasite. The parasite enter into the red blood cell, and then they start making their own protein and express and insert it into the surface of the infected cells. These proteins, which are produced and exported onto the surface of the infected cells, they are called variant surface antigens. And one of these variant surface antigens is PFAMP1, which is expressed on the surface of the infected red blood cells. PFMP1 is Plasmodium falciparum erythrocyte membrane protein 1, is encoded by a family of genes called the VAR genes, and there are 60 copies of this gene in haploid genome of the parasite. And each of them are expressed in mutually exclusive fashion. Only one is expressed at a time. This is to show you, This is to show you the distribution of the genes in the 14 chromosomes of the parasite. Mostly the VAR genes are located in the subtelomeric and telomeric ends of the chromosome. Based on their location in the chromosome, the direction of their transcription, which is indicated by the arrows, and the complexity of the protein, which is encoded by these genes, VAR genes has been classified into 3 major groups. The type A VAR genes, APS A, the type B VAR genes, and also the type C, which are indicated in blue. A closer look into the VAR genes. VAR genes have 2 exons. Exon 1, which is the bigger one, encodes for the extracellular domains of PFMP1. The exon 2, which encodes for the intracellular domain. Today I will be focused on the 3 domains which are encoded by the exon 1. Now, these domains are highly polymorphic, and the genes which codes for these domains are very diverse. So based on the classifications that we have, PFMP1s can be grouped into 3 general groups, the type A, the type Bs, and the type Cs. Each of them have a characteristic combination of domains of Duffy binding-like and CIDR, cysteine-rich interdomain region domains. So the type A's are characterized by DBL alpha 1 and CIDR alpha 1, whereas the type B's and C's are characterized by DBL alpha 0 and CIDR alpha 226. So I'll be focused on these domains. That's because these domains has been shown to be important in the parasite pathogenesis. To look at the function of these proteins, which are encoded into the surface of the infected red blood cells, they are essential for the parasite to make them sticky. Once red blood cells are infected, they become sticky and they can bind to endothelial cells. This enables the parasite to avoid a splenic clearance. But for the host, the binding of these infected red blood cells to the endothelial cells is a challenge. That's because when there is sequestration of infected red blood cells in the blood vessels, they can block blood vessels and leads to complications of malaria, such as cerebral malaria. The other essential role of these PFMP1 is because they are encoded by a family of genes, one gene is expressed at a time in a mutually exclusive fashion, so that these parasites are capable of antigenic variation. What you see here is the parasite expresses a certain variant type, and then they switch into a different variant type, and then they keep switching. This switching of variant types, which are expressed on the surface of the infected cells, enable the parasite to escape from the host immune response. Despite all this variation, PFMP1s has been shown to be the major targets of naturally acquired immunity. In addition to their diversity and differences in the domain compositions, PFMP1s has also been associated with different infection outcomes. The type A PFMP1s has been associated with binding to endothelial protein C receptor, intercellular adhesion molecule 1, chemokine receptor 1, and others, and which has been linked with severe malaria, which is the cause of up to 600,000 deaths every year. The other type of PFMP1 is the VAR2CSCA, which binds to the chondroitin sulfate A receptor in the placenta, and has been associated with sequestration of parasite in the placenta and pregnancy-associated malaria, which is responsible for the deaths of 100,000 infants and more than 10,000 mothers every year. And the other type of PFMP1s is the type Bs and the type Cs. They've been linked with mild malaria. This specialization in the functions of PFMP1 opens an idea of having a syndrome-specific vaccine, which is based on PFMP1, because PFMP1s are major targets of immunity, but they are really diverse. But they also show specialization in function. So there is a possibility to develop vaccine, for example, to pregnancy-associated malaria, which is based on VAR2CSA, and also vaccines for severe childhood malaria, which is based on the type A PFMP1s. So this leads me to the aims of my PhD project. My PhD project was focused on understanding how diverse these genes are. The gene which encodes PFMP1s, there are 60 genes per haploid genome, but they undergo mitotic and meiotic recombination. So in the field, in the real setting, how diverse is this gene with a focus on the genetic diversity in PNG, and looking at how different are parasites in terms of their virgins in different parts of Papua New Guinea. And the second part of my PhD is to understand naturally acquired immunity to PFMP1 variants with a focus on understanding the pattern and hierarchy of antibody acquisition to this PFMP1 in young Papua New Guinean children, and identifying those PFMP1s which are associated with clinical outcomes, especially those which are associated with severe and clinical malaria. And we are also interested in looking at the dynamics of antibody response in severe childhood malaria. So first, I'll go through the phylogeography and genetic diversity of the virgins. What you see here is globally the diversity of virgins has been shown to be spatially different. For example, what you see here is the y-axis is unique types, unique VAR types. The x-axis is the number of VAR sequences. In Africa, if you keep sequencing a new sequence, the probability of getting a new type is very high. All the sequences are very different. But when it comes to, at least in one site in PNG and also in Brazil, what was observed is, as you keep sequencing, your probability of getting a unique type that you've already seen is very high, suggesting that the diversity in these regions are limited as compared to Africa. But within PNG, we know that there is a difference in the transmission of malaria. There is difference in terms of the human population and the vector species. So we wanted to further look at the diversity of virgin in two high transmission areas of Papua New Guinea. So we've sampled two regions. One is in the East Sepik province, and the other one is in the Madang province. So the Wosara village here, and also Migul, which is in the Madang province of Papua New Guinea. We've sampled 33 samples from Wosara, from 6 different villages, and 35 samples from Migul, from 4 villages. And then we compared what we found from these villages with the previously published data set that we have from PNG. In order to do the diversity, we use the DBL alpha domain. Why we are using the DBL alpha is because the DBL alpha is the only domain which is found in almost all virgins. So we can use it as a marker of diversity. So we amplified block B to H of this gene using degenerate primers. These degenerate primers are capable of amplifying as much possible virgins. So then we separated these different amplicons by cloning them into a topo vector. And then I picked 96 colonies for each sample, which is a total of 6,528 colonies, which were sequenced. So we analyzed the sequences, and then we used the high-quality sequences to determine the diversity and geographical differences between populations. What we found here is, again, these are the unique types in the y-axis, and the x-axis is the number of sequences. As we keep sequencing in two other different regions, what we found is there is spatial difference in the diversity of virgin in PNG. It's not similar everywhere. In some areas, it's low, as observed before, but in these areas, it's higher than what we found, what was reported from the previous region. This can be related to the transmission intensities, differences in the vector species, and differences in the human population and things like that. But when we look at how many of the virgins are identical in these 3 different regions, what we found is very striking. Only 9 genes are 100% identical in these 3 regions, suggesting that the VAR types are evolving in different regions separately. Then we compared the observed and estimated diversity of these genes between Africa and Papua New Guinea and Brazil. What we found is the overall diversity in PNG is 5 to 8 times lower than the one which is estimated from Africa. The African sites are mainly in Kenya, Senegal, and Gabon. So overall, the overall diversity of virgin in PNG is limited. But we know that there is a spatial differentiation in the diversity of virgin. Then we looked at how similar the virgins, how similar are isolates or samples which are collected from the same region in terms of their var types. What we see here is this is the relatedness of the parasite based on their geographical origin. This tree shows that the red ones are those samples which are collected from Wasara, and the blue ones are collected from Migu. What we found is those samples which are collected from Wasara, they are more related in terms of their var types than a sample which is collected from a distant population, suggesting that virgins show strong population structuring in Papua New Guinean populations. Then we looked at a fine scale level, because we have 6 villages from Wasara and 4 villages from Migu. Do we see this difference at a fine scale level? And then we investigated the similarity of the relatedness of samples based on their var types. What we found is very striking at this distance. That's because those parasite populations that we collected from these different regions, they are different in terms of var types. And those virgins which are collected from the same region, those samples which are collected from the same region, they are more related in terms of their virgins. So we were saying that virgins can show a fine scale population structuring in Mughal village. This is in line with different barriers. One of the barriers between these villages, even if they are very close to each other, there are differences in the human population. There are cultural and language barriers. The people don't marry each other, even if they live very close to each other in terms of these villages. And also we have differences in the vector species which transmit malaria in the coastal region and inland villages. And also we have well-documented microepidemiological variation in PNG at village level. But when we look at the Wasara village, we don't see this structuring. All parasites from the different villages are similar in terms of their relatedness based on the var types. This can be explained by the way of life in Wasara. This is a highly densely populated populated area in PNG, and migration and circulations are a way of life. And also there is an overlap in the vector species which transmit malaria, and if you see the distance between the villages is not very high. So with this, I'll try to summarize the first part of my talk, which is I've showed you that the diversity of virgin in PNG is moderate as compared to Africa, and there is a spatial heterogeneity within PNG. It's not similar and homogeneous everywhere. And we found that virgins can be potentially used as a marker of recent population structuring. This is a kind of model that we put. The isolates which are collected from this region are different from the isolates which are collected from this region. But within the Wasara, we don't see any clustering, but within Mughal village, we see parasite clustered based on their relatedness of the virgins. And these studies enabled us to sample a large number of unique DBL clones, which can be used for the investigation of antibody responses to PFMP1 variants. So this leads me to the second part of the talk, which is different from the first one, but it is on naturally acquired immunity to PFMP1 variants. We know that we have a conclusive evidence now. Naturally acquired immunity to malaria can protect from severe disease and clinical disease and death, but sterile immunity is never fully achieved. But kids under the age of 5, they are fully susceptible for both clinical malaria and severe malaria. But as they get older, their susceptibility to severe malaria starts to decline, and their susceptibility to clinical malaria also declines by the age of 25. But sterile immunity is never fully achieved. We'll find people who have parasites in the blood, but they don't have any clinical symptom because they develop immunity to the clinical disease. So our focus is we wanted to understand the naturally acquired immunity in the age group, in the younger age group before the age of 5, where they are very susceptible to clinical and also severe malaria. Acquisition of naturally acquired immunity has been well studied in PFMP1, and there are different proposals. This is one of the proposed epidemiological feature of antibody acquisition to PFMP1 types. What you see here is in the right y-axis is antibody level from low to high, and in the x-axis is the age. The one which is indicated in orange is the pattern of antibody acquisition to those PFMP1s which are associated with severe malaria. And the one you see in the red is those PFMP1s which are associated with uncomplicated malaria. So what we see is maternal antibody starts to decline, and then the child starts to make their own antibodies to those PFMP1s which are associated with severe malaria, the type A PFMP1s, whereas slowly they acquire antibodies to the type Bs and the type C PFMP1s. So in this gap, there is a gap as the maternal antibody starts to decline, and until they get their own antibody level higher, there is a gap. And at this gap, the disease risk is very high, and the parasite is very virulent because the immune system is very low in these age groups. So we wanted to focus on this age group and understand the acquisition of immunity to PFMP1s, type A PFMP1, type B and C PFMP1s, and identify which PFMP1s are associated with protection from clinical outcomes in this age group. Just to remind you, this is the blood stage life cycle. In the blood stage, antibodies can be acquired to the merozoites, which can block invasion of red blood cells. And antibodies can be acquired to those antigens which are expressed on the surface of the infected cells. These antibodies, which are acquired to the antigens which are expressed on the surface of the infected cells, has been shown to be important. They can disrupt cytoadhesion, which is shown in vitro. They can disrupt rosetting. Rosetting is the adhesion of infected cells with non-infected cells. They can also block adherence, and although there is no very conclusive evidence, there are a few studies which show reversal of adhesion. And animal model studies also showed that antibodies can enhance phagocytosis by opsonization. So antibodies which are acquired to the variant surface antigens, the proteins of the parasite which are inserted on the surface of the cells, are functional, and they are important in protecting, in preventing cytoadhesion and preventing clinical symptoms of malaria. But the challenge with this kind of studies is how can we account for the antigenic diversity and the antigenic polymorphism in these genes. It's really diverse gene. As we saw, we cannot even count the total number of genes in the field. How can we account for this antigenic diversity? And it's a major challenge in defining the antigenic diversity of this gene. To address this challenge, our approach is mainly focused on 3 domains. One is the DBL alpha domain, the CIDR alpha domain, and the DBL beta domains. These domains are important ligands of the parasite to bind to different host cell receptor, which leads to cytoadhesion or leads to sequestration and rosetting. So in the first part of the talk, I've showed you that we've looked at the genetic diversity. The idea is not just only study the genetic diversity. The idea is mainly identify the unique types which are circulating in the field. And then we identify these unique circulating types, then we'll have a sequence library where we have a larger number of DBL clones, unique clones from the field, and also we have NTS DBL, NTS CIDR domains. We included a larger region of the domain because especially the NTS domain has been shown to affect the protein structure in the DBL domain. Then what we did is after we did a sequence-specific PCR to each groups, we amplified the whole genes for NTS to CIDR, and also the DBL clones from the field isolates. And then the proteins are expressed by the cell-free RTS expression system, and they are printed into a protein array. In addition to that, we have functionally distinct PFMP1 domains, the NTS DBL, DBL beta, and CIDR domains. And these domains were expressed in the conventional E. coli-based expression system. So these proteins allow us to control the protein expression in the cell-free expression system. And then we measured antibodies to many of these antigens by different platforms. We measured antibodies to the protein array by a protein array serology screening method, and also we measured antibodies by Eliza, Luminex, and CBA in collaboration with other labs within Australia and outside of Australia. In order to do the serology, in order to test the antibodies, we have antibodies which are collected, or plasma samples which are collected from the field. We used a cohort of sample from the Eliza cohort. Before I start describing the epidemiological and clinical outcomes of this cohort, I will use the following 3 terms very frequently in the next part of my talk. Molecular force of infection. When I say force of infection, it is the number of new clones that a child acquired during in that follow-up period. And the other term is clinical malaria. When I say clinical malaria, it is the presence of fever, an auxiliary temperature of greater than 37.5, and parasite density more than 2,500 parasites per microliter. And we have severe malaria cases. When I say severe malaria, it's according to the WHO guideline. They have a clinical malaria with complications such as impaired consciousness, respiratory distress, severe anemia, and others. So this cohort, the Eliza study, was conducted just here, just above one of our study area. In this study, 264 children with a median age of 1.7 years, they were enrolled into the study, and then they were followed for 16 months. The kids were observed every 2 weeks to check for clinical symptoms, and every 8 or 9 weeks, a sample was collected, a finger prick blood sample were collected to check the presence or absence of parasites by microscopy and PCR. So we know how many clinical episodes they have during the follow-up period, how many infections they had, when they were infected, and with which species. So in total, we have 28 severe malaria cases, and each child has, in average, 6 new infections during the follow-up period, and at least 2 clinical malaria during the follow-up period. So this kind of cohort studies enable us to ask question, which type of antibodies, the antibodies that we measure at this point, whether they are associated with protection from severe malaria or protection from clinical malaria. So we did serology on about 800 samples, 233 samples at enrollment, and 233 samples at the end of the follow-up. And in addition to that, we have these severe cases. We measured antibodies before, during, and after the severe malaria. But for today, most of the data that I'm showing you is based on the analysis of the data at enrollment. We measured the antibodies at enrollment, and we analyzed those data. So why are we focused on these 3 domains? Is because these domains are essential ligands of the parasite. One of them is DBL1 alpha. I'll talk about the DBL alpha domain. The DBL alpha domain has been classified into 6 groups based on the number of cysteine residues that they have. The cysteine residues are indicated in red box, and also the sequence signature in the position of limited variabilities. We have these 4 positions of limited variability. How many cysteine residues do they have, and what sequences do they have in this position of limited variability has been used to classify this DBL alpha sequence into 6 groups. This classification also matches well with the type A, the type B, and the type C classification that I've showed you. So the group 1 and 2 and 3, they have 2 cysteine residues, and they are mainly type A type DBL alpha domains, whereas the group 4 and 5, they have 4 cysteine residues in the sequences, and they are DBL alpha sequence of type B and type C PFMP1s. whereas the group 6, they have either 1, 3, 5, or more than 5 cysteine residues in the sequences. So we use this classification to classify the DBL clones that we have on the protein array. So this is the proportion of the different groups in the protein array. We have 456 of them. Of these, 84 of them are cis2, group 1, 2, and 3, and we have 341 cis4 and 31 cis-X type of DBL domains. So we measured antibodies using this protein array. What you see here is a protein array probed with an Australian adult plasma sample, which is our negative control. So there is no much reactivity to our proteins. And then we probed it with Papua New Guinean children with a lower level of exposure. When you see the reactivity, they have antibodies to many of the DBL types, but they still have gaps in their antibody level and their reactivity. And this is a Papua New Guinean adult. What you see here is the red is more reactivity. So we have higher reactivity to the antigens in the Papua New Guinean adults. So this showed us that there are gaps in the Papua New Guinean children in terms of their reactivity to the different DBL clones. So I'll show you what we found in terms of the antibody data from these protein arrays. So the y-axis here is the mean intensity, and the x-axis is the different groups. In each group, we have 233 children. They are sorted by their age, from the smallest age group to the highest age group. So what we see here is there is reactivity. Overall, there is higher reactivity to the cis2 domains. And for group 1, group 2, and group 4, there is a statistically significant increase with age in terms of their antibody level. So this is not a new thing. These have been reported before. So this is similar to the previous reports. Overall, the kids recognize 17% of the kids have antibodies, detectable antibodies, for 17% of the group 1 and 25% of the group 2s. But for the group 3, 2 to 6, the reactivity is low. So now on, I will use this classification based on the number of cysteine residues. Group 1, 2, and 3, they have 2 cysteine residues, and I'll call them cis2. Group 4 and 5, they have 4 residues, 4 cysteine residues. I'll call them cis4, and the group 6 is the cis-X. So when we compare between the groups based on their cysteine residues, there is high reactivity to the cis2s as compared to the cis4 and the cis-X, in terms of both the magnitude of the antibody level and in terms of the breadth, how many of the DBL clones they recognize. So there is these children under the age of 3 years, they can recognize many of the cis2 VAR types. And then we looked at if these antibodies increase with exposure. So first, we wanted to define what exposure is. In this case, what we define exposure is the total number of infections that they have throughout their lifetime. We're trying to have a kind of proxy. It's because we didn't follow these kids from birth to the age of 3. We have a kind of proxy. We measured the number of infections that they have during the follow-up period. Then we multiplied the number of infections that they have during that follow-up period times their age, which is to find an overall exposure throughout their lifetime. And then we grouped them into those kids with low exposure, medium exposure, and high exposure. So the type 2s and the type 4, the cis2s and the cis4, they show a statistically significant increase with exposure. Such association with increase in age and infection status has been reported before. And then what's interesting here is because we have 3 different sites, because we've sampled 3 different sites. One is the Wasara, which is very close to the Eliza cohort study where our plasma sample is collected. You can see this is the plasma sample collection site. And this is where we have the DBL clones. and we have DBL clones from these further sites too. And then we saw reactivity. If there is a cross-reactivity, these kits should be able to recognize all the DBL alpha clones from the different regions. But based on our observation from the genetic diversity, we've seen VAR types from different areas are different. So we wanted to look at how these kits cross-react with VAR types which are collected from different geographical locations. What we found is overall, these kits significantly, there is higher level of recognition to those DBL clones which are collected from the local population, suggesting that the kits acquire antibodies to the local circulating parasites. But the overall reactivity to those DBL clones which are collected from the different geographic regions is very low, 10% for Miggle and 5% for Amale. And then we looked at in the different classification level. Why we are classifying them is because the type A PFMP1s are relatively conserved, so there should be more reactivity for the type A or the cis2 type, DBL alpha between different geographic regions. But what we found is there is cross-reactivity, but the level of cross-reactivity is very low. The kits recognize up to 40% of the cis domains, which are the cis2 DBL domains, which are collected from this region, but they don't recognize the overall reactivity is less than 20% and 10% for Amale for those which are collected from a different geographic region. But we don't see that difference for the cis4. This probably is because the overall reactivity of the cis4 is not very high, and cis4 is extremely diverse. And then, okay, these kits acquire antibodies to these different domains. Are these antibodies protective? How are they associated with clinical outcomes? So what we did here is we measured the antibodies at enrollment, and then we grouped them based on the level of the antibodies. This is a typical graph that you get if you graph the overall antibody reactivity in a population. Some have higher level of antibodies, some medium, some have low level of antibodies. And then we grouped them into 3 equal groups so that we can compare those with high level of antibodies versus those with low level of antibodies in terms of the number of clinical episodes, in terms of the number of severe cases that they have during the follow-up period. So we used negative binomial regression to determine that. We calculate the incidence rate ratio. The incidence rate ratio of clinical episodes for those kids with high level of antibodies versus those kids with low level of antibodies. If the incidence rate ratio is 0, that is 100% reduction in risk. If it is 0.5, that's 50% reduction in risk. And if it is 1, that means there is no change in risk. If it is more than 1, there is an increase in risk. And in all graphs that I'm going to show, I'll put the 95 confidence interval. But in this cohort, we know that there is variation in clinical malaria with age. It varies with age. It varies with the number of infections that they have, and it shows variation between different villages because malaria is very heterogeneous. In some areas there is high transmission. In some areas there is a low transmission. So village of residence of the kids affects the number of clinical episodes that they get. And also, we have a variation in terms of season. Malaria is a seasonal disease. And also, we have factors such as the length of the follow-up period for how long these kids are followed for, and the prior use of anti-malarial drug, and the usage of bed nets. These all factors can affect the number of clinical episodes that they get. And also, when we measure the antibodies, infection status at the time of antibody measurement can also affect the level of the antibodies. Those which have a concurrent infection usually have higher level of antibodies. So we have to account for all these differences when we look at the association of antibodies with clinical outcomes. So all the incidence rate ratio that I'm going to present, they all are adjusted for all these factors. So I call it as adjusted incidence rate ratio. So when we look at this here what you see here is the y-axis is the incidence rate ratio and 95 confidence interval, and the x-axis is the different groups. So of all these 6 groups, we only have group 2, which is associated with reduced risk of clinical episodes. High level of antibodies to group 2 domains is significantly associated with reduced risk of clinical malaria. And then when looked at high-density clinical malaria, just increasing the parasite level. So with those kids with high-density parasitemia, again, we see a significant reduction in the risk as compared to those kids which have low level of antibodies, suggesting that the group 2 DBL alpha domains are significantly associated with a reduced risk of clinical malaria. But some of PFMP1, such as the group 4 domains, they are associated with increased risk of clinical malaria. And then in this cohort, we have kids that we measured antibodies at enrollment, and then they were followed for 16 months. And these kids were followed 16 months. After 16 months, some of the kids acquired severe malaria. So then we grouped them into two, those kids which had severe malaria during the follow-up period and those kids with no severe malaria. But when you look at the age and the number of infections that they have during the follow-up period, it is comparable. So how is the antibody level different for these two groups in terms of the antibodies measured at enrollment? So we looked at the antibody levels. So those kids which got severe malaria, indicated in red, had significantly lower level of antibodies as compared to those kids which didn't get severe malaria, suggesting that those kids with severe malaria had gaps in their antibody repertoire to the cis2 PFMP1s. But when we look at the antibody level at the end of the follow-up, after they had about five infections and a severe episode, we don't see that difference. So at this point, the level of the antibodies in those kids with non-severe malaria and those kids which got severe malaria is similar, suggesting that the gaps in the antibody repertoire are filled during the follow-up period when they had these repeated infections and severe malaria. And then when we closely look into the different groups, the antibody level in these groups, those kids which had severe malaria and non-severe malaria, when you look at the antibodies, we see significant difference in the mean reactivity. For example, if we look at group 1 and group 2 and group 4, we see those kids with severe malaria had significantly lower level of antibodies as compared to those kids with non-severe malaria. So this raised the question, can we predict severe malaria, whether kids are going to get severe malaria or not, based on their antibody level to group 1 and group 2? And we did this receiver operating characteristics curve, which shows the sensitivity versus the specificity. So the area under the curve is indicated here for all the groups. If the area under the curve is 0.5, that means these antibody levels are not predictive. There's no prediction. If it is 1, it's like a perfect prediction. So what we found is for group 1 and group 2, there is about 0.84 for group 1, and for group 2, it is about 0.8 prediction accuracy. So suggesting that low level of antibodies to group 1 and group 2 can predict whether kids are going to get severe malaria or not. So the other thing is, I've showed you these before. These are the proposed pattern of antibodies to type A PFMP1 and the type BC PFMP1s. So this is what we observed in PNG. This is a type A, the characteristics of type A PFMP1s, the characteristics of type B and cis PFMP1s. So you see that the antibody level in the proposed pattern, the antibody level peaks at the age of 3. What we found here is also the same. The antibody level to the type A PFMP1s or the cis2 domains peaks by the age of 3. So these simultaneous measurement of antibodies to these PFMP1 variants can enable us define the age-dependent pattern of antibody acquisition to PFMP1 types. This leads me to the other domain, which is the CIDR domain. The CIDR domain is another interesting domain because it's been shown to be a ligand for endothelial protein C receptor, CD36, CD31, and IgM. This domain has been classified into 3 major groups. Those CIDR domains which bind to EPCR, they are classified as EPCR binders, and those CIDR domains which are associated with rosetting, but we don't know their receptor yet. They are classified as rosetting-associated CIDR domains, and the CIDR alpha 226, they've been shown to bind to CD36 binders. So we measured antibodies to these domains, 19 APCA binder domains, 4 rosetting domains, and 12 CD36 binders. We measured antibodies at enrollment, before, during, and after severe malaria, and at the end of the follow-up. So today I'm showing you only the enrollment antibody level. So at enrollment, what we found is, here is the mean intensity of the antibody levels and the different CIDR domains. The red ones are APCA binders, the yellow ones are CD36 binders, the grays indicated here are CIDR deltas and gamma. So the red dotted line is the median of overall reactivity to the 35 domains. What we found is there is no much difference in terms of reactivity, whether the CIDR domains are APCA binders or CD36 binders. When you look at the seroprevalence, the overall seroprevalence is pretty much comparable, but when you look at the top 10, 6 out of the top 10 are APCA binders. Then we looked at whether these antibodies are associated with, again, clinical outcomes. What we found is antibodies, high level of antibodies to the APCA binders are significantly associated with reduced risk of high-density clinical malaria, but we don't see that association in low-density clinical malarias. Then we looked at the DBL beta domain. The DBL beta domain is one of the interesting domains because there are only 2 DBL beta domains which are able to bind to ICAM1, intercellular adhesion molecular 1. The binding of infected red blood cells with intercellular adhesion molecular 1 has been associated with cerebral malaria, although it's not very conclusive. So we looked at the antibody acquisition and the association of antibodies to this domain with protection. So antibodies were measured for 5 domains. These domains, NTS, DBL, CIDR domains as a control. What we found is there is high reactivity, like 40% of the kits have antibodies to DBL beta domains. And then we looked at whether these kits with high level of antibodies are protected as compared to those kits which do not have antibodies. What we found is, again, it's interesting that a high level of antibodies to the DBL beta domains has been significantly associated with reduced risk of high-density clinical malaria. The red one is clinical malaria with more than 10,000 parasites, whereas the black one is clinical malaria with more than 2,500 parasites. It's a borderline for the clinical malaria with 2,500 parasites, but we've seen significant reduction in the risk of clinical malaria in those which have high level of antibodies. So to summarize the overall association studies, association of antibody level with protection, here is indicated clinical malaria, low density. When I say low density, it is greater than 2,500, high density greater than 10,000 parasites per microliter, and severe malaria. And also this is increased risk. This one is reduced risk of clinical malaria. So the group 1 DBL alpha, the group 2 DBL alphas are significantly associated with reduced risk of low-density clinical malaria, whereas the group 2, the CIDR alpha 1, and also DBL2 beta, they are significantly associated with reduced risk of high-density severe malaria, high-density clinical malaria. Group 1 and 2, and also CIDR alpha 1 and DBL2 betas are significantly associated with reduced risk of severe malaria. But some of the antigens, such as the group 4, CIDR alpha 2.9 and CIDR alpha 3.3, they are associated with increased risk of clinical malaria, showing that high level of antibodies to these domains might be associated with high level of exposure. So with respect to these 4 domains, group 1 and group 2, CIDR alpha 1 and DBL2 beta, which are associated with severe malaria, I'll show you the antibody levels of protected children versus kids which got severe malaria. So these are the antibody levels indicated here. Green is low level of antibody, Black is medium level of antibody, and red is high level of antibodies. And with respect to the group 1s, the group 2, CIDR alpha 1, EPCA binders, and DBL2 beta. These are 24 children with severe malaria. So all of them are either low or medium. They have low or medium level of antibodies at enrollment. But when you look at kids which are protected, when I say protected, those kids which have no clinical malaria during the follow-up period. So when you look at those kids with no clinical malaria, we see at enrollment, they have high level of antibodies to DBL alpha 1, group 1 and group 2, and also CIDR and DBL2 beta domains. But if you see some of them have low level of antibodies, but they are still protected, suggesting that these are just not the factors which protects from severe malaria. But what's interesting here is this probably shows that there is an additive and synergetic effect of antibodies to these different domains to protect kids from severe malaria. So to summarize what I showed you today, so the diversity of Varg in PNG is moderate, and it shows strong geographical differences, and antibodies are acquired to the local DBL alpha domains. Type A PFMP1s, in terms of both CIDR alpha 1 and also the cis2 DBL alpha domains, are major targets of the PFMP1 domains. And high level of antibodies to the group 2 domains is strongly associated with protection from clinical and severe malaria. But interestingly, this group is not very diverse. All studies which are published till today, they only showed 5 to 10% of the DBL sequences are under the group 2. So the number of group 2 sequences are really low, suggesting that these sequences are less diverse and are also associated with protection. And gaps in antibody repertoire to PFMP1 domains are associated with severe malaria. And these gaps are filled after the severe malaria or after having 5 infections. This leads me to thank people who are involved in these studies. First of all, I would like to thank the children, guardians, and the communities who are involved in these studies. And I would like to thank my supervisors, Eliza, Ivo, and Diana. They've been very supportive, and their office is always open for me to ask questions and get help. I would like to thank the both Barry and Mueller lab members. They are really nice people to work with and very supportive at times of difficulties during the PhD. Specifically, I would like to thank Stephanie Monk. She did some of the work that I haven't discussed today. Diga expressed some of the protein, and I've used sequences which are developed by Abby in collaboration with the Malaria Gene Network, but I haven't presented it today. From Mueller Lab, I'd like to thank Cornelia Weisman. She's been very supportive in terms of the statistics and the statistical analysis of my data. Michael White, he analyzed some of the data I haven't presented today. And also Camila, we work in a very close project, so for all the discussions. I would like to thank everyone in the infection and immunity division, particularly Clara and Tony. They've helped us in expressing the proteins. And Chris Chu with the CBA assay. And John, she's always there to keep me organized. I would like to thank my PhD committee, Louis, Tony, and Freya. They've been very supportive during my studies. I would like to thank Doug. He supported me and Danica to go to PNG and visit all the villages where the samples are collected. Actually, that's when I start to trust the data that I've seen. I would also like to thank the Protein Array Lab in the UCI, University of California, Irvine. I would like to thank Philip Felgner, Ray, and Al for all the work that they did to express the proteins. It's not easy to express 543 proteins. Queensland Institute of Medical Research, I would like to thank Denise. She hosted me for a month in her lab to do the protein array serology, and she introduced me to the concepts of these protein arrays. I would like to thank Thomas Lavesen and Thor Theander at Center for Medical Parasitology in Copenhagen. They hosted me for a month and I did Luminex serology in collaboration with them. And they are fantastic people to work with. I would also like to thank Jacob and Bent. They did a lot of work in extracting sequences from the whole genome, which I haven't presented it today. And the ICAM1 project, it was done in collaboration with people from Florida Atlantic University, mainly Andrei Olinikov and Olga. I would like to thank Papua New Guinea Institute of Medical Research, Peter, Siba, Livingston, Joe, and Benson, and Enamor. They're involved in one or another way in the collection and processing of the samples. And Nicholas Sen also involved in the collection of samples. And John Reeder, who was involved at the early stage of this project. Finally, I would like to thank my families, friends, and my wife, Enu. I would like to thank the University of Melbourne for my scholarship, and this project was supported by NHMRC, and I would also like to thank the Aussie Muller and the Australian Society of Parasitology for supporting me to go to Copenhagen, and I would like to thank STMH too. Thank you very much. Okay.
Thanks. That's a really nice, comprehensive piece of work you've done there. That you should be really proud. I had some questions about the analysis towards the end that you presented there. So you showed lots of associations, and the way that you analyze data, we're looking at individual different domains.
Yeah.
I was wondering if you managed to do some analysis to try and kind of tease out the relative contribution of antibody responses to those domains to protection against severe disease. Because I imagine some of them might be quite closely correlated.
Yeah. Actually, in the first part, in the DBL alpha tab, what I showed here is a multivariate analysis where we included the antibody level of each groups. It's adjusted for the overall antibody level of the groups. But for the CIDR domain, again, for the EPCRs, it is summarized by functional groups because it has 35 domains. We summarize them into 3 groups, the APCR binders, CD36 binders, and rosetting. And then each of them were adjusted separately for the antibody level. But what I want to do is combine all of them and then see which one is really important. That's a really good idea, and I'm planning to do that. Yeah.
Well, it's probably a ridiculous question because it refers to about your 5th or 6th slide, I think. But there was, when you were talking about the number of variants of PFBMP1 in Papua New Guinea and how they were related to the intensity of transmission and comparing that to Africa, you had a graph that had on it very high bars which were called estimated and other bars which were called observed. And the observed didn't look very different between New Guinea and Africa, but the estimated did. I was wondering if I looked at that right, what that meant.
Yeah, actually, if you just see the bars, it doesn't seem very different. But what's interesting is, okay, if you have 800 sequences, and if you get 700 unique types, almost you're getting 1 type per sequence. But in PNG, what you usually get is half. If we sequence 800 sequences, we'll get 400 types. The number of unique types that we're getting in PNG is very low, relatively lower as compared to Africa. I'm not saying the diversity of variation in PNG is low. It is a relative term. It's still high, but as compared to the overall diversity in PNG is lower.
So you saw association of protection with some of the FAR domains, but then you also saw association with increased risk.
Yeah.
So can you speculate on why antibodies to certain FAR domains are going to increase the risk of severe disease?
It is not, the antibodies are not increasing the risk. Rather, it is the antigens are markers of increased risk. That's because, for example, if we see group 1 and 2, they are cis2 type PFMP1s, and cis2 type PFMP1 or type A PFMP1s, they are expressed, they've been shown to be expressed in younger age groups, and they are associated with severe malaria. So being high level of antibodies to those and being protected, it makes sense. But the other ones, the cis4s, they are really diverse, and if you have high level of antibodies to these, it shows that you are getting more infections. That's why you are getting high level of antibodies. Yes.
I'm interested to know if you can estimate the fold difference between low antibody levels and high antibody levels in your signature and your responses to the different VAR epitopes.
It's all relative because the antibody measurement for group 1, group 2, and group 3 is different. What we see, when we group them high, medium, low, it's all relative for that specific group.
Sure, but, and as a consequence, is it twofold or fourfold or tenfold between low and high?
So if we take the lowest and the highest, the difference can be, for example, 10,000 MFI. The mean fluorescence intensity can be 10,000 the highest, and the lowest will be 100 or 200. So there's a lot of difference when we look at the actual fluorescence intensity. But in order to standardize the analysis between groups, what we do is we take relative classification to classify them as high in that specific group, medium in that specific group, and low in that specific groups.
The reason I'm asking is because whenever you've got an antibody response to a multivalent surface, as you have, then quite often really quite small differences in concentration of the antibody can make a big difference in the binding. So I was curious whether even the protection that you're seeing might be evoked by quite small differences in antibody level.
The antibody level, if you see the actual numbers, so the thing is, what I'm showing here is, one is it's normalized for all, so the analysis between different platforms is a bit different depending on the platform.
Understood.
Yeah. So what I show here is, it is for the protein array, the difference seems, for example, 10.1 and 10.2. That difference in terms of MFI is 100 MFI difference. But it's presented in logarithmic scale.
And how would that translate into an antibody difference or concentration difference?
So I haven't looked into a concentration of the antibodies. Yeah, that's an interesting thing to look at in terms of quantifying how much is the concentration. Yeah.
Questions? Okay, well, 3:05, so finished there. Thank you, Sofonias.